So in this video, I'm going to look at cell division and how is it important to human body or to an eukaryotic cell. Kindly like and subscribe to the channel as we proceed with the tutorial. So what then is cell cycle? When we say cell cycle, then you're making reference to the series of events that take place in an eukaryotic cell that leads to its replication. So this cycle occurs mainly or only to an eukaryotic cell because eukaryotic cells have membrane-bounded organelles, or they have their nuclear materials defined in the cytoplasm, unlike the prokaryotic cell. So the series of events that take place in organism that leads to its replication is what we call the cell cycle. So the cell cycle have two main stages. Before a cell can divide, it has two main stages. The first stage is what we call an interphase, and the second stage is what we call a division phase. So we have the interphase and the division phase. So let's look at the interphase, then we proceed with the division phase. When we say the interphase is basically the resting stage of the cell. So before the cell can divide, it must undergo some resting stage and then synthesize several or some molecules that is needed for its synthesis. Example of these molecules are the enzymes, the proteins, the RNAs, the DNAs, and the ribosomes. So all these molecules are being synthesized at the interphase, and it's called the resting stage, and it's also the longest stage of the cell cycle. The interphase has three main stages, and each of these stages have a particular reaction that occurs at the stage. We have the gene 1 phase, we have the S phase or the synthetic phase, then the gene 2 phase. So these are the series of events that occurs at the gene 1 phase. So at the gene 1 phase of the interphase, the cell grows in size and chemical changes occur in the cell. RNA and DNA and ribosomes and several enzymes are synthesized as earlier. Then during the same phase 2, the cell prepares itself for the cell division. The S phase of the interphase, which is also the next stage of the resting stage, is the synthetic phase. The synthetic phase is where the proteins and the enzymes which are required for the cell division is synthesized. So here the cell synthesizes proteins and the enzymes that are synthesized, that are needed for the synthesis or the replication of the cell. Also, the cell duplicates its chromosomes. The chromosomes are needed for cell division because they carry the nuclear or the chemical genetic materials of the cell. So at the S phase or the synthesis phase, chromosomes are also synthesized or duplicated to its sister chromatid. The gene 2 phase is the stage of the interphase at which, or let's say, is between the completion of the DNA synthesis and the beginning of that. So this one is just the end of the interphase. So the first phase of the cell cycle, the interphase, is where the cell prepares itself for the next phase. It synthesizes several organisms or several molecules which are required for the synthesis or the duplication of the cell. The next phase of cell cycle is the cell division, and the cell division is basically the division of the cell itself. The cell will divide by either the nucleus or by the cytoplasm. We have two main types of the cell division. We have the mitosis and the meiosis. So in this video, we are going to stick to the mitosis that involves the division of the cell or the somatic cell that results in the maintenance of the number of chromosomes in the daughter cells. So for the mitosis, the number of chromosomes are maintained in the daughter cell, but unlike the meiosis, which occurs in the reproductive cells of the gonads, the number of chromosomes are halved. So here we are going to look at the mitosis and the various stages involved in the mitosis. So the mitosis is basically the type of cell division in which single parent cell is divided into two daughters such that the number of chromosomes in the daughter cells are maintained. So here the number of chromosomes in the daughter cells are maintained, unlike the meiosis. So the cell division of the mitosis also has two main stages. We have the cytokinesis and the karyokinesis. So before a cell can divide, the nucleus must divide first, then the cytoplasm divides. So the division of the cytoplasm is what we call the cytokinesis. Then the division of the nucleus is what we call the karyokinesis. So let's look at the division of the nucleus. The division of the nucleus or the karyokinesis also has four main stages. And the four main stages of the karyokinesis are the prophase, the metaphase, the anaphase, and the telophase. And each of these phases have different, chemical reactions or chemical change that occurs. For the prophase, the chromatin material become condensed, shortened, and coiled. So the condensed form of the chromosomes is what we call the chromatin. So the chromatin become condensed in the nucleus. It becomes shortened and it becomes coiled. Then the two sister chromatids attach with each other at the centromere to form a thread-like chromosome. So if you look at this picture, we have this one as a centriole, which is an organ that emerges during cell division. We have this one, the two strands of the chromosome are what we call the chromatids. And these ones are merged at a point we call the centromere. So the centromere is a point of attachment of sister chromosomes or sister chromatids in the chromosome. The next phase of the karyokinesis is the metaphase. And the metaphase is very simple. Spindle fibers emerges from the centrioles. So during cell division, basically, centrioles emerges at the opposite poles of the cytoplasm. Then the centrioles begin growing a microtubule, which is called the spindle fiber, that leads to the disintegration of the nuclear membrane. So the nuclear membrane disintegrates or disappears and allows way for the spindle fiber to attach the sister chromatids or the chromosomes in the nucleus. That is the metaphase. Then at the anaphase, the sister chromatids of the chromosomes are being split to the opposite poles of the cell. So one centromere is here, one centrole is here, and the other centrole is here at the anaphase. They grow a spindle fiber and attach to the centromere of the chromosomes. So when it attaches at the anaphase, it splits the sister chromatids to the opposite poles. So one goes here and the other goes here. So this is the late or the early anaphase. Then at the telophase, the spindle fibers break down because the sister chromatids have been splitted already. Then the nuclear material reappear, whereas the nuclear membrane is formed. So when you split it, and each of these chromatids are moved to the opposite poles of the cytoplasm, the nuclear membrane is formed back with the same number of chromosomes in each daughter cells. Then the last stage of the cell cycle, the cell division, is the cytokinesis, which involves the division of the cytoplasm to form the two daughter cells. So that's how basically cell division occurs. So these are some significance of mitosis. Mitosis is used for growth. It's important in replacement of cells. It is used for asexual reproduction. It aids in regeneration. Then also it helps in healing of wounds. So for growth, for multicellular organisms to grow new cells, it is mitosis that divides or that ensures the division of the somatic cells to produce new cells that replaces the old or dead cells. So that's how basically cell division occurs in a cell. Kindly like